Welcome to the physiology class. My name is Radhika P R. Today we are going to discuss about composition, function, regulation and applied physiology of salivary glands. In humans, saliva is secreted by three pairs of major salivary glands and some minor salivary glands. Major salivary glands are parotid gland, submaxillary or submandibular glands and sublingual glands. The minor salivary glands include lingual mucus gland, lingual serous gland, buccal gland, labial gland and palatal gland. And next we are going to discuss about the major salivary glands in detail. The first gland is parotid gland. Parotid glands are the largest of all salivary glands. These are situated at the side of the face, just below and in front of the ear. Each gland weighs about 20 to 30 grams in adults. Secretions from these glands are emptied into the oral cavity by means of Stenson duct or parotid duct. This duct is about 35 to 40 mm long and opens inside cheek against upper second molar tooth. The second major gland is submandibular gland. It is otherwise known as submaxillary glands. These glands are located in submaxillary triangle medial to mandible. Each gland weighs about 8 to 10 grams. Saliva from these glands are emptied into the oral cavity by Watson duct, which is about 40 mm long. The duct opens at the side of frenulum of tongue by means of small opening on the summit of papillae called carungula sublingualis. The next major salivary gland is sublingual gland. Sublingual glands are the smallest salivary glands situated in the mucosa at the floor of the mouth. Each gland weighs about 2 to 3 grams. Saliva from these glands is poured into 5 to 15 small ducts called ducts of Ravinus. These ducts open on small papillae beneath the tongue. On the other duct is larger and it is called Bartholin's duct. It drains the anterior part of the gland and opens on carungula sublingualis near the opening of submaxillary duct. And these diagrams showing the acini and duct system in the salivary glands. Salivary glands are again classified into three types based on the type of secretion. These are serous gland, mucus gland and mixed glands. Serous glands are mainly made up of serous cells. These glands secrete thin and watery saliva. Parotid gland and lingual serous glands are also serous glands. Second type is mucus gland. Mucus glands are mainly made up of mucus cells. These glands secrete thick viscous saliva with high mucin content. Lingual mucus glands, buccal glands and palatal glands belongs to this group. And another category is mixed gland. Mixed glands are made up of both serous and mucus cells. Submandibular, sublingual and labial glands are mixed glands. Salivary glands are formed by acini or alveoli. Each acinus is formed by small group of cells which surround a central globular cavity. The central cavity of each acinus is continuous with the lumen of the duct. The fine duct draining each acinus is called intercalated duct. Many intercalated ducts join together to form intralobular duct. Few intralobular ducts join to form interlobular ducts, which unite to form the main duct of the gland. A gland with this type of structure and duct system is called racemose type. Next, we are moving on to the properties of saliva. Per day, there is about 1000 to 1500 ml of saliva secreted, which is slightly acidic in nature with a pH of 6.35 to 6.85. Next, we are moving on to the composition of saliva. Saliva is composed of 99.5% of water and 0.5% of solids. Solids includes both organic and inorganic substances. The organic substances include the enzymes like amylase, it is otherwise called thiolin, then maltase, lipase, lysozyme, phosphatase, carbonic anhydrase and kallikrein. Other organic substances include mucin, albumin, proline-rich protein, lactoferrin, immunoglobulin A, blood group antigens, free amino acids, non-nitrogenous protein substances like urea, uric acid, creatinine, xanthine and hypoxanthine. Inorganic substances include sodium, calcium, potassium, bicarbonate, bromide, chloride, fluoride and phosphates. Saliva also contains small amount of oxygen, carbon dioxide and nitrogen. Next, we are moving on to the functions of saliva. Saliva is a very essential digestive juice since it has many functions. Its absence leads to many inconveniences. The first function of saliva is preparation of food for swallowing. When food is taken into the mouth, it is moistened and dissolved by saliva. The mucous membrane of mouth is also moistened by saliva. It facilitates chewing. By the movement of tongue, the moistened and masticated food is rolled into a bolus. Mucin of saliva lubricates the bolus and facilitates swallowing. Second function is digestive function. Saliva has three digestive enzymes, namely salivary amylase, maltase and lingual lipase. Salivary amylase is a carbohydrate digesting enzyme. It acts on cooked or boiled starch and converts it into dextrin and maltose. Though starch digestion starts in the mouth, major part of it occurs in stomach because food stays only for a short time in mouth. Optimum pH necessary for the activation of salivary amylase is 6. Salivary amylase cannot act on cellulose. Another enzyme present in the saliva is maltase. Maltase is present only in traces in human saliva and it converts maltose into glucose. The next enzyme is lingual lipase. Lingual lipase is a lipid digesting enzyme. It is secreted from serous glands situated on the posterior aspects of tongue. It digests milk fats. It hydrolyzes triglycerides into fatty acid and diacylglycerol. The third function is appreciation of taste. taste is a chemical sensation. By its solvent action, saliva dissolves the solid food substances so that the dissolved substances can stimulate the taste buds. The stimulated taste buds recognizes the taste. Fourth function is cleansing and protective function. Due to the constant secretion of saliva, the mouth and teeth are rinsed and kept free of food debris, shed epithelial cells and foreign particles. In this way, saliva prevents bacterial growth by removing materials which may serve as cultural media for the bacterial growth. The enzyme lysozyme of saliva kills some bacteria such as Staphylococcus, Streptococcus and Brucella. Proline-rich proteins present in the saliva possess antimicrobial property and neutralizes the toxic substances such as tannins. Tannins are present in many food substances including fruits. Lactoferrin of saliva also has antimicrobial property. Proline-rich proteins and lactoferrin protects the teeth by stimulating enamel formation. Immunoglobulin IgA in saliva also has antibacterial and antiviral actions. Mucin present in the saliva protects the mouth by lubricating the mucous membrane of mouth. Another function of saliva is role in speech. By moistening and lubricating soft parts of mouth and lips, saliva helps in speech. If the mouth becomes dry, articulation and pronunciation become difficult. The next function is excretory function. Many substances, both organic and inorganic, are excreted in saliva. It excretes substances like mercury, potassium, iodide, lead and thiocyanate. Saliva also excretes some viruses such as those causing rabies and mumps. In some pathological conditions, saliva excretes certain substances which are not found in saliva under normal conditions. Example is glucose in diabetes mellitus. And in certain conditions, some of the normal constituents of saliva are excreted in large quantities. For example, excess urea is excreted in saliva during nephritis and excess calcium is excreted during hyperparathyroidism. The next function is regulation of body temperature. In dogs and cattle, excessive dripping of saliva during panting helps in the loss of heat and regulation of body temperature. However, in human beings, sweat glands play a major role in temperature regulation and saliva does not play any role in this function. And the last function is regulation of water balance. When the body water content decreases, saliva secretion decreases. And this causes dryness of the mouth and induces thirst. When water is taken, it quenches the thirst and restores the body water content. That's all about the functions of saliva. Next, we are moving on to the nerve supply and regulation of salivary secretion. Salivary secretion is regulated only by nervous mechanism. Autonomic nervous system is involved in the regulation of salivary secretion. Salivary glands are supplied by both parasympathetic and sympathetic divisions of autonomic nervous system. First, we are going to discuss about sympathetic nerve supply to salivary glands. Sympathetic preganglionic fibers of salivary glands are arises from lateral horns of first and second thoracic segments of spinal cord. The fibers leave the spinal cord through the anterior nerve root and end in superior cervical ganglion of the sympathetic chain. Postganglionic fibers arises from this ganglion and are distributed to the salivary glands along the nerve plexus around the arteries supplying glands. And when these nerve fibers are stimulated, less amount of thick saliva rich in mucus is secreted. Next is about the parasympathetic nerve supply to all the salivary glands. Preganglionic nerve fibers arises from superior and inferior salivatory nucleus. From this nucleus, preganglionic nerve fibers arises. Nerve fibers arising from the superior salivatory nucleus where terminates in submaxillary ganglion. Postganglionic fibers arises from this ganglion, terminates on sublingual gland and submaxillary gland. The nerve fibers arising from inferior salivatory nucleus terminates on otic ganglion. Postganglionic fibers arises from this ganglion, terminates on parotid gland. When it's stimulated, large amount of watery saliva secreted with less amount of organic substances. Next is about the mechanism. Salivary secretion is regulated by nervous mechanism that may lead through reflex action. That includes conditioned and unconditioned reflex. First, we are going to discuss about unconditioned reflex. Unconditioned reflex is the inborn reflex which is present since birth. It doesn't need any previous experience. When we place food in the mouth, automatically salivation occurs. So, next we are moving on to explanation of this part. When any substance is placed in the mouth, the taste buds are getting stimulated. That send impulses to the salivary centers. Thus, inferior and superior salivatory nucleus are getting stimulated. Impulses from these glands are finally reach the salivary glands and salivation occurs. The next reflex is conditioned reflex. Conditioned reflex is the one that is acquired by experience and it needs previous experience. Presence of food in the mouth is not necessary to elicit this reflex. Sight, smell, hearing or thought of food are the stimuli for this reflex. And these stimuli send the impulses to higher centers like cortex and hypothalamus. Stimulated higher centers send the impulses to salivary centers. These centers send the impulses to salivary glands and finally salivation occurs. That's all about the regulation of salivary secretion. This is the schematic diagram showing the salivary secretion. This is also another schematic representation. Next, we are moving on to the applied physiology. That includes hyposalivation and hypersalivation. And what is hyposalivation? Reduction in the secretion of saliva is called hyposalivation. It is of two types, namely temporary and permanent hyposalivation. Temporary hyposalivation occurs in emotional conditions like fear, and fever, dehydration, etc. Permanent hyposalivation occurs in obstruction of salivary ducts, congenital absence, Bell palsy, etc. And what is hypersalivation? Excess secretion of saliva is known as hypersalivation. Physiological conditions when hypersalivation occurs is pregnancy. Hypersalivation in pathological conditions is called thialism, sialorrhea, sialosis, etc. Hypersalivation occurs in the following pathological conditions also, like decay of tooth, disease of esophagus, neurological disorders, nausea and vomiting, etc. Other disorders related to the salivary glands are xerostomia, that means dry mouth, and drooling. Drooling means excessive salivation or dribbling of saliva, and Sjogren's syndrome. It is an autoimmune disorder. That's all about composition, function, regulation, and applied physiology of salivary glands. Thank you.